This is the fourth part of my video, organization and expression of immunoglobulin genes, and I had to stop last time because my video was 10 minutes already, so I'm just going to repeat it again. In this video I'm talking about the mechanism of how the recombination happens and these RSS segments are actually deleted. So I have here a VK gene and a JK gene. There are more VK and more JKs, but I'm just not showing them. So if they are arranged this way versus this way, the first thing is that they are always oriented by the recombinase enzymes. So the recombinase enzyme, they make this VK and JK oriented, and I'm just using the kappa chain as example. Same thing happens in lambda gene and same thing happens in heavy chain variable region. Now after this, what happens is that it's like one from right at the junction of this my gene and the RSS, one of the strand is cut. So this is how it would look like. So I have this, and so you guys could see like one strand is cut, and then I have this, and so one strand is cut, and this is again done by the RAG1 and 2 RAG2 enzymes. These enzymes cut them off. So same thing happens over here too. I'm just drawing small pictures because I didn't have so much space, and I'm using ball pen now. Same reason. So one strand is cut. That's our second step. First step is just a rearrangement, and second step is one of the strand right at the junction of the gene and this RSS. This is also called coding segment, and this is called our coding sequence and signal sequence. You guys might see it in the book. So coding sequence is the actual gene, and signal sequence is the RSS. So right at the junction of this coding sequence and signal sequence, cut is made in one of the strands. And after this, what happens is, let's look at this one. So we have nucleotides, like we have nucleotides here. So rather than like this nucleotide being like by itself and this nucleotide is actually like continuing with this one, that's why I have this. One of these attacks the other one and they make a hairpin structure. So this is how it looks like after this stage. So one nucleotide attack the other one and we get a hairpin like this, because the nucleotide here now makes a bond with this one, and our DNA is not like this but it's a hairpin. And for this RSS, like both the strands here, they are just like blunt like this because they are just cut. There's no hairpin right here. So it goes up, and again this is cut, and for the RSS it's just like blunt ends, nothing's there. But for our coding sequence, it makes up this hairpin. Same thing happens right here. So we have this, makes a hairpin right here. Again it makes a hairpin right here, and this RSS just comes off, and we have these like blunt ends right here. And this reaction is again catalyzed by this RAG1 and RAG2 proteins. Now what happens after this structure is, I'm not going to draw this box for a second just to show what happens. So let's say for example I'm drawing this right here, and I just have this like hairpin right here. So what happens after this step is, this hairpin is cut at random position. Let's say this hairpin is cut here by endonuclease enzyme. By endonuclease, it's cut randomly, and what happens is that this opens up, right? Now the top one is long and the bottom one is short. It happens like this. And after this, like, nucleotides are added over here to make it longer, and this cut happens randomly. It could happen right here, right here, right here, right here, anywhere. And the enzymes are added, and you can see also that this step, it adds diversity to the variable region. This whole thing we're talking about is variable region, and these things adds diversity to the amino acid coded by variable region. And these nucleotides that are added are called P nucleotides, and they are called P because they are palindromic, and palindromic because they read both on same side. For example, I'll give you guys an example. So let's say we have this, and let's say we have ATGC, ATA. So our bottom strand would be TACGAT, and this is hairpin right here. Yep. So now let's say that the hairpin is cut right here. So what happens after this is ATGCTA, and coming this way TAGC, and right here we have DA, and the new nucleotides that are called P nucleotides are added over here, and this would make it CGATA and TCG. So if you look at this part, because it was cut right here, I'm going to draw a line right here. So if you look at this part is palindromic. So if I read it this way, GCTATAGC, GCTATAGC, this is palindromic. That's why these nucleotides are called P nucleotides. So after this step, what happens is that it's cut open. It becomes like one is longer, other one is shorter, or they could be equal length too, and the P nucleotides are added. And also some of these nucleotides, after being added, some of these are chopped off after this by some of these are chopped off. Actually I take it back, I'm not so sure if they are chopped off, but these P nucleotides are added for sure. Now after this, and this thing only happens on heavy chain, some more nucleotides are added after this, and this only happens on heavy chain, and these like new added nucleotides are added randomly, and they are called N nucleotides. For example, let's say I add ATZ and it will be TAC. So these nucleotides, like these added ones, they are called P nucleotides, and these new added ones, they are called N nucleotides. And let's see what happens after this. So in our picture, in this we'll have this. We have a blunt end right now. We don't have a hairpin anymore, and nothing happens to the RSS. Same is how it is. And we have this, and a blunt end at the both ends, and same thing happens over here. And after this, another enzyme called double strand. This is a group of enzyme called double strand break repair, DSBR. BR enzymes, they ligate like these two strands, like they are gonna ligate. So we will have, this is our VK from the beginning, and then comes our JK, now this, and this is our added P nucleotide. And if this was our heavy chain instead of light chain, we'd also have our N nucleotides right here. So we just created a diversity right here, and this happens randomly. and so these two are joined, and what happens is like this region it falls off. So we have this one RSS, the second RSS, and it falls off just as a double stranded stuff. And for this one the same thing happens. I'm running out of time, so I'll tell you. So first these two are created bond and by addition of P and N nucleotide, but in this case how they are joined is this comes and joins right here, and this it goes and joins right there. So the back part of this RSS goes and joins here. So we have this, and this one is in the beginning, and this one is this one, and so this one, we have this one, and then we have P or N nucleotides here, and then we have this one right here. So this is how we completed the recombination. Thank you for watching my videos. Keep watching more. Bye.